Here's something that might completely change how we think about the origins of life. Scientists just created a molecule so unstable, it's practically explosive. Yet it could hold the key to understanding how life began in the universe. A groundbreaking international research collaboration has achieved what many thought impossible, successfully creating methanetetrol in laboratory conditions for the very first time. This remarkable feat was accomplished by a diverse team of experts. Ryan Fortenberry, who specializes in astrochemistry at the University of Mississippi, Ralf Kaiser, a distinguished chemistry professor from the University of Hawaii at Manoa, and Alexander M. Mebel, a computational chemistry expert at Florida International University. Their extraordinary findings have been published in the prestigious scientific journal Nature, marking a significant milestone in our understanding of prebiotic chemistry. But here's where it gets fascinating and controversial. Fortenberry describes this compound as essentially a prebiotic concentrate, a seed of life molecule. To help beginners understand this concept, imagine you're looking at the very first ingredient that could eventually lead to the complex chemistry we associate with living organisms. As Fortenberry eloquently explains it, think of it like an acorn that will grow into a tree in the grove. The acorn alone cannot make a tree. It requires sunlight and water, and lots of other things. But it can be what starts the process. This analogy beautifully illustrates how methanetetrol might serve as a fundamental building block, requiring the right conditions and additional components to eventually contribute to life's emergence. Now, here's what makes this discovery particularly intriguing and technically challenging. Methanetetrol belongs to a notoriously elusive family of compounds called ortho acids. These mysterious substances have long frustrated researchers because they're incredibly difficult to isolate and study under normal laboratory conditions, despite scientists believing they play a crucial role in the chemical processes that preceded life on Earth. To recreate the conditions where this molecule might naturally form, the research team had to think like cosmic engineers. They designed an experiment that mimics the harsh environment of outer space by freezing a mixture of water and carbon dioxide to temperatures approaching absolute zero. That's nearly negative 273 degrees Celsius or negative 459 degrees Fahrenheit for perspective. Then they bombarded this frozen mixture with radiation similar to cosmic rays that constantly travel through space. This extreme procedure allowed them to liberate the methanetetrol molecule as a gas, which they could then identify using powerful ultraviolet light detection methods. Kaiser emphasizes the groundbreaking nature of this achievement. The detection of the only alcohol with 4 hydroxyl groups at the same carbon atom pushes the experimental and detection capabilities to the final frontier, the next level beyond what could be accomplished before due to the lack of experimental and computational approaches. For those unfamiliar with chemistry terminology, hydroxyl groups are combinations of hydrogen and oxygen atoms, and having 4 of them attached to a single carbon atom creates an extremely unusual and unstable molecular structure. And this is the part most people miss. Methanetetrol is essentially a molecular time bomb. The compound is so incredibly unstable that it will decompose rapidly unless researchers maintain it under precisely controlled conditions. This instability arises from its unusually high concentration of oxygen bonds, and here's why that matters. Oxygen atoms naturally resist forming bonds when they're in close proximity to other oxygen atoms, making such molecular structures energetically unfavorable and prone to breakdown. Fortenberry offers a vivid description that makes this concept accessible to everyone. You have this compact carbon-oxygen molecule that just really wants to go boom. And when it does, when you give it any kind of energy, you'll have water, hydrogen peroxide, and a number of other potential compounds that are important for life. It is like a prebiotic bomb. This explosive analogy might sound dramatic, but it perfectly captures the molecule's tendency to rapidly transform into other compounds that are fundamental to biological processes. But here's where the controversy begins, and where this research becomes truly thought-provoking. The research team argues that since they can create this molecule in laboratory settings, the same processes could theoretically occur in the vast expanses of space. This reasoning opens up fascinating possibilities for astrochemists who are actively searching for environments that might harbor the conditions necessary for life to emerge. Fortenberry presents a compelling argument that expands our perspective on potential life-supporting environments. While carbon is the building block of life, oxygen is what makes up nearly everything else. Oxygen is everywhere and is essential for life as we know it. So, if we can find places where methanetetrol forms naturally, we know that it is a place that has the potential building blocks to support life. This statement suggests that discovering methanetetrol in space could serve as a cosmic signpost, pointing us toward regions where life might be possible. However, this raises some provocative questions that deserve serious consideration. Are we perhaps overstating the significance of creating one unstable molecule in highly controlled laboratory conditions? Does the ability to synthesize methanetetrol under extreme artificial conditions really translate to meaningful insights about life's origins in the universe? Some skeptics might argue that we're making dramatic leaps from laboratory curiosities to profound statements about life itself. Moreover, there's an interesting philosophical dimension to consider. If this molecule is so unstable that it immediately breaks down into other compounds, is it truly a building block of life, or is it more accurately described as a transitional step that quickly leads to the actual building blocks? The distinction might seem subtle, but it could significantly impact how we interpret these findings and allocate resources for future research. The financial investment in this research shouldn't be overlooked either. This groundbreaking work was made possible through funding from the National Science Foundation, specifically Grant AST 2403867. This raises important questions about research priorities. Should we be investing heavily in creating highly unstable molecules that exist for mere moments? Or would those resources be better spent on more direct approaches to understanding life's origins? The complete research findings have been published in a detailed study by Marks, J.H., and colleagues in Nature magazine, with the full citation available as doi.org/10.1038 SS41467-025-61561Z. This publication represents years of collaborative work and sophisticated experimental design, making it a significant contribution to the scientific literature regardless of how one interprets its broader implications. What do you think? Does this discovery represent a genuine breakthrough in our understanding of how life might emerge in the universe? Or are we reading too much into the successful synthesis of one extremely unstable molecule? Are the researchers justified in calling methanetrol a seed of life? Or is this an example of scientific overreach? Should taxpayer-funded research prioritize these types of fundamental investigations? Or would we see better returns from more applied research approaches? Share your thoughts. Do you believe this research brings us meaningfully closer to answering the profound question of how life begins? Or does it simply add another interesting but ultimately inconsequential data point to our scientific knowledge base?